I was just kidding. My name is Mr. Nicholas, but I like to be called Dr. Nicholas, or just simply the scientist. So today, if you've noticed, I have some of our world's favorite fruit on the table, right? We have the family of the strawberries, right? We have the Papa strawberry, who's full and healthy and ripe. Just look at them. You can just red and seeds everywhere on the outside. And you have the Mama strawberry, who's just beloved and nurturing, right? However, today, our problem is the younger strawberries, right? The children. We got Mike and Tina. Oh my God. If you take a look at the children's strawberries, they aren't as ripe and strong and healthy as their family is, right? The Papa strawberry and the Mama strawberry. So today, guess what? We're gonna get all of our stress out with our fingers, take out the DNA from the strawberry. And when I mean take out the DNA from strawberry, I mean take out the DNA from the strawberry. What is DNA? Forgive my manners, right? We have deoxyribonucleic acid, which is simply instructions that's way deep inside of you that you get from your parents, mom, and pops, right? As simple as that. However, in strawberries, it's a bit different. Strawberries are technically octoploids, and which means that they have more sets of DNA from their parents, which ultimately means they have instructions on top of instructions that's just gonna leak out into the instructions for the children's strawberries are quite complicated, right? And if you notice, they aren't the same size, so it is my job and your job, our job, to get to the bottom of this, right? We're gonna take those instructions, really take a hard look at these instructions and say, I wanna figure you out. We're gonna have full and more healthy strawberries at the end of the season, right? And I promise you that.
Problem: the young strawberries are not growing as healthy as the parent strawberries. Experiment. Let's do an experiment. Extracting DNA from the young strawberries will help us understand what is wrong with their instructions. Here are the materials and supplies required to conduct this experiment.
So, first thing first, what you will need is a Ziploc bag, a common Ziploc bag, right? If you do not have a Ziploc bag, plastic bag may work or may not, it's part of experimenting. We have household soap, liquid soap. We have isopropyl alcohol, 70% rubbing alcohol. Again, I advise anyone who's doing this project to do it with a partner, an older guardian, or someone who can guide you guys through any step you're confused on. And salt.
Here is a step-by-step preview of what we will be doing today.
So for today's purposes, we will remove the stem from the strawberry, right? Maybe asking, why are we removing the stem from the strawberry? Well, it's quite simple. We don't have the materials or the tools necessary to extract the DNA from the strawberries, right? We're gonna keep the parent strawberries alive and well as they are. Just look at them. So, first thing first, it's very important. You have your strawberries, make sure the Ziploc bag is completely sealed at the top. Now, this is my favorite part of the experiment. You kind of get a second to, smooth these without the ice, Just get a good mash. We're gonna mash the strawberries up all the way until, this may take 2 minutes, maybe more minutes, depending on how big your hand is. And that's fine, right? Strawberry no longer. Now, it's very important at this part to have your household materials. We have a 1/4 cup of water, right? It's very important. It's just water. You can get out your sink, you can get out of the bottle, it doesn't matter. We're gonna get the teaspoon of salt. Just enough, right? We're gonna do a teaspoon of salt. We're gonna do a teaspoon of liquid detergent. You may be asking, what does these salt and soap has to do with anything, right? So basically what we creating is a lysis, right? Which is a solution used to separate the membrane, the DNA from the cell itself, right? Because inside the cell is more than the DNA, but instead we specifically looking for the instructions, like I said earlier. Thirdly, we will open up the bag, and guess what? You got it. Gently pour our solution, our lysis, in the bag of mashed strawberries. Now, this step is very important. Because we have liquid inside of the bag, we don't want to mash, right? Instead, what we can do is gently just like massage the bag. So what we're doing is we added the solution to break down the cells. I'm taking the whole strawberry itself and making it easier for the instructions to creep out. You may can see some of the instructions. Nah, I'm just kidding. You can't see the instructions yet because we're waiting for a step that's very important. That's called precipitation, right? This is the moment where you'll see the actual instructions removed from the gush. So now we're getting very close to our goal of the day, right? And that's just removing the instructions from the baby strawberries that aren't growing so ripe and healthy. Our next step will be to simply filter the strawberry gush, right? So I like to use a napkin. If you have a coffee filter, that's awesome. But a napkin will do, right? You open up the napkin until you get the 4 boxes, right? It's simple. So we're gonna take this filter of this 4 square opened up napkin and we're gonna just place it over a clean cup. Be careful. I advise you guys to use gloves, latex gloves, whatever you can get your hands on, right? So you may ask a friend to hold down the napkin, but if you're doing this yourself, it's okay because so am I. So we're gonna pour our strawberry solution inside of the filter, right? And it's very important that the napkin doesn't fall inside and that we actually do not get none of the larger parts inside of the cup. So this process may take overall about 2 to 5 minutes depending on how much you're pouring and depending on your filter. But it's important to get most of your solution into the filter, back into the clean cup. It's okay to have leftovers. Finally, we're gonna be very careful. This is probably the most important part, is to not get any of the big chunks inside of our clean cup of instructional strawberries. Notice how I perfectly wrapped around the strawberries. You can still get enough out of the filter just by simply pressing onto it. Cool? All right, cool. Last but not least, we have our precipitation, which ultimately you will pour the alcohol inside of our solution of strawberry and salt and soap until we actually see the instructions come out of the strawberries. I kid you not, this is magic. So here we go. You can pour, they say to pour as much as strawberry solution you have, but for today's purposes, we will finish the bottle, I should say. So you want to gently just stir picture the web. It should be sort of kind of a web, you know, the instructions. It's just so many. You ever open up a how to fix a couch? Yeah. Ooh, this is tricky, good stuff right here. Wow. So what you have here is the DNA of the baby strawberries. And our goal, like I said, is to package this up nice and neatly so it doesn't get any more germs on it. Send it over to a team of professionals who will take care of our instructions and thoroughly go through them and read the instructions and figure out what is causing the baby strawberries not to grow. Again, as promised, we grabbed the instructions from the baby strawberry DNAs. And what I can also promise you is that the DNA that's inside you and I are all the same. It will look just like this. Okay, now this last step is not only for myself, but I encourage you all to clean up after yourself, thoroughly wash your hands, sing the birthday song as you're washing your hands, and as promised, the parent strawberries will be in good shape.